Edson. I'm the agronomist and laboratory specialist here at Brookside Laboratories in Western Ohio. We're joined today by Dr. Andrew Margenot from the University of Illinois. Just real quick, briefly on Dr. Margenot's background. He's completed his PhD at UC Davis in soils and biogeochemistry just a couple of years ago, 2016. His work focused on organic matter cycling, enzymes, and phosphorus cycling in East Africa. He began his position as an assistant professor of soil science at Illinois in Champaign there in August of 2017. And his current laboratory program works is integrating enzymes into biogeochemistry, as well as soil phosphorus management, tropical perennial crops, and then just more directly to working with Brookside Laboratories. We've been fortunate enough to work with Andrew on some projects he's got going on, and we hope to further that relationship now with some of these enzymes that he's going to be talking about today. We'll be introducing and offering to our consultant network here in the very near future. We've run a lot of internal work here, but nothing that we've put on a commercial scale for folks to start using yet. And I'm happy to see some folks with the NRCS here. And with that, I'll pass it on to Andrew. Like I said, I'll be muting all mics, so please just type something in the comment chat bar, and at a proper time to interject, I'll disrupt Andrew and get some interaction going. So thanks again for joining us this morning, everybody. And Andrew, the floor is yours.
Thank you, Chris, and thanks for having me. Thank you for being here. So as Chris said, I want to take the next, well, I think we've got 90 minutes, an hour and a half to go over some discussion on enzymes. And as Chris said, it sounds like we can field questions as I go through this. Chris, I'd be more on the let's answer questions as we go side of things. I'd like for this to be more of a discussion. So with that said, I'd like to just dive in. The basic purpose of this talk is to share with you what my group has been doing for about 2 years now, pretty intensely on enzymes. As a bit of background, I've been doing enzyme assays for 5 or 6 years now, not so long. The technique is actually 50 years old this year, or rather the first application of enzyme assays to soils is 50 years old. The first time that was done was in Iowa in 1969. So my 5 years is not very long, but we've come across in our lab some questions that began as honest questions of, hey, what do we do to run this assay for this sample type? And that has, as you all probably well know, that kind of leads to this rabbit hole of methodology type questions. So I want to share with you what we have been finding on what do we do with, say, pH or buffer. What do we do when it comes to practical questions of enzyme assays, like do we use 37 Celsius? Do we use X amount of substrate? There's also some conceptual questions buried in this. So with that said, I'm gonna go ahead and let's talk about enzymes. So the way that I think of enzymes is that they have high potential, but we haven't really tapped into that potential as a community of scientists, especially in soil science. The reason why enzymes, I think, have high potential is that they do all the work that I think we give microbes a little bit too much credit for. We talk about how microbes decompose organic matter, and technically it's not the microbes, it's the enzymes made by the microbes. Now, I know that might sound like a technicality, but as we'll see, the fact that these enzymes are outside of the microbial cells, so they're extracellular, that has profound implications for how we can measure those enzymes that catalyze decomposition. And it also has implications for how enzymes play a role in nutrient cycling. So enzymes have high potential because they do the work of decomposing. And at the same time, the fact that these enzymes are largely extracellular, so existing outside of the cells in which they were expressed, that also makes them even more potentially powerful because they can integrate abiotic and biotic. So abiotic things like minerals, because, for example, enzymes can bind to minerals. They also are sensitive to temperature, to pH. So by virtue of being outside of cells, they're even more fine-tuned to the environment, as well as what we do when we manage soils. Now, this is a bit of a catch-22 because by virtue of being outside of the cell, you also open up the enzymes to lots of potential artifacts when it comes to how we can measure them. So that then is the catch-22 of these enzymes being extracellular, is that they are challenging to quantify. And there's two levels of this. One is how we perform assays in a way that we can have confidence in the data. And the other aspect of challenging to quantify is how we quantify them in a way that gives meaningful data, conceptually speaking. So if enzymes are outside of cells, what are we really measuring if we air dry the sample versus keep it fresh? We know that with microbiomass, we need to run it on field fresh samples. We want living cells. But if enzymes are not in cells, what does it mean if we're measuring it on air dried samples? Maybe it is okay because there's no cell implication in that enzyme's activity. Okay. So let's talk about why enzymes, why would you care about enzymes, even if you're not into the biogeochemistry and the potential of them? Well, more practically, they've been proposed as a indicator of soil health. So here is a shot from the NRCS website. Enzymes are one of the recently proposed soil health indicators. And if we look at the soil health technical note that was posted about a year ago, this note was part of a federal registered comment that was proposing to codify or to canonize specific indicators of soil health. If you dive into this document, which I had a lot of fun doing, and you read the fine text, what my understanding is that the 6 processes that were identified have concurrent indicators. And if there's an NRCS person who would wish to correct me, please jump in. And from what I can tell is enzymes are situated in this NRCS framework as the process of general microbial activity. So the idea is that enzymes give us a window into how active these microbes are. The document then goes on to lay out 4 specific enzymes in soils. And so these are highlighted here in bold: beta-glucosidase, glucosaminidase, also known as chitinase, and then acid or alkaline phosphomonoesterase, also called phosphatase, and final asulfatase. So we have an enzyme that does carbon. Nitrogen and carbon is the chitinase, phosphatase is phosphorus, sulfatase is sulfur cycling. We also see.
I'm sorry. Andrew, just to chime in on that. So those are based on that document and some work that we've done here and other stuff. Those specific 4 enzymes are the ones that I mentioned earlier that we've been doing internal working with, and those are the ones that we'll be offering here at Brookside soon.
Oh, great. So those 4, Chris, are also featured in the Soil Health Institute's website. And so if we look at another source of, well, which enzymes are thought to be or being proposed as being useful for soil health, well, here's a different perspective. I want to point out, though, that I think it's meaningful that the Soil Health Institute lists enzymes as a tier 2 indicator. So tier 2 indicators, from what I've gathered, are largely the biological ones, which are defined as needing additional research before we can really frame them or interpret them with more rigor. So a tier 2 indicator, that's in contrast to a tier 1 indicator of soil health. Again, this framework is not from NRCS, it's from the Soil Health Institute. And if we look at their website of the tier 2 indicators, which are basically a highly promising but needs more work type indicator, what we find is that those same 4 enzymes are popping up again. Just to I think it's important to keep in mind that these 4 enzymes that are being talked about are just 4 of many different types. Here is another depiction of common enzymes. Here they are being categorized by the nutrient elements that they catalyze the degradation of. So with yellow stars, I've indicated the 4 enzymes that are being considered by NRCS, by Soil Health Institute, and it seems like also by Brookside Labs. But keep in mind that there are other enzymes out there, and even this is also underestimating it. If you want to think about how many potential enzymes there might be in soils, I suggest that you crack open a textbook of human biochemistry, and they'll give you this, very complex pathway of, for example, amino acid breakdown. And every step in that chain of 50 arrows and boxes is effectively an enzyme catalyzed step. So in soils, there's probably thousands and thousands of different enzymes, both a different class of enzyme, so it breaks down a kind of bond, but also different kinds of isozymes. So isozyme with a Z is a different flavor of enzyme that does the same job. And this gets into some isozymes are better or less able to get at different kinds of substrates. So for example, with phosphatase, is that phosphoester bond embedded in a DNA or in a lipid compound? Well, that difference would be dictated by the isozyme potentially. So we're going to focus on these 4, or rather think about these 4 as we go forward. Not sure if that was a question or just background.
That was my background.
Oh, no worries. Okay. Well, speaking of that, any questions so far? Okay. I want to go over some basic principles of enzymes. So I teach a grad course on enzymes, and we always give about a week and a half, sorry, the first 2 weeks rather, to just discuss what enzymes are. So forgive me if you know this, but I think it's good to go back to the basics. And this is a theme that we'll talk about when it goes to running enzyme assays in soil samples, is that I think we would do well as a community to revisit some of the basics, both the basic tenets of biochemistry and enzymes, but also some of the very first papers that were written on soil enzymes. And again, that's relatively recently. The 1960s was the emergence of the assays of enzymes, at least with artificial substrates in soils. So let's talk about what enzymes are exactly. I'm sure that you've all heard about it, but to get really specific, enzymes are catalysts. They're also called bio-nanocatalysts. And they're called biocatalysts because, well, they're made of biomacromolecules. Specifically, they're made of proteins. And proteins are these long chains of amino acids shown on the left. Now, the definition of a catalyst is that it increases the kinetics, but doesn't change the thermodynamics of a reaction. So what that means is that you can go faster. Now, whether that reaction happens is a different question, right? That's the question of thermodynamics. A good example is that something is favored thermodynamically, but it might take 1,000,000 years to happen, a slow trickle. Enzymes come in by expediting that process. They speed it up. And the reason why enzymes are vital for all life forms is that without enzymes, we would have to rely on diffusion and on thermodynamically favorable, but not necessarily kinetically okay processes. So things for cells have to happen quick. Enzymes were evolved to deal with that. They make things happen quickly. Now, the other part of the definition of a catalyst is that the catalyst is never consumed in the reaction. So that means that a small bit of enzyme or of a catalyst goes a long way. You just need a pinch of it because it keeps being reusable by the substrates or by the reactants to make more product. Once it makes product, that enzyme goes back to the beginning of the reaction and makes more and more product. So they're very, very powerful. This just gets into more nitty-gritty on how catalysts work, but think of it as catalysts lower the activation energy. In the case of enzymes, they oftentimes will bend the substrate, and by bending it, they favor certain bonds to be broken more readily. And so that's why enzymes are able to lower that barrier to that process going forward. Hence, they speed things up. In soils, what enzymes are catalyzing are the decomposition of organic matter bonds into, well, they hydrolyze those, and those result in the inorganic nutrient element form. So specifically, we're thinking about organic nitrogen, phosphorus, sulfur, and carbon all being hydrolyzed into the inorganic forms. Or in the case of carbon, polysaccharides becoming glucose, and then the microbial cells take over from there. Mentioned how enzymes are proteins, and proteins are a pearl string necklace of amino acids. Well, think of it almost as a pearl that is magnetic, because those pearls on a string can then interact with each other, and from that you get different levels of structure that arise. So you have primary structure, simply the sequence of amino acids. Secondary through quaternary structure arises from hydrogen bonding and other interactions amongst those amino acids. The result is that you get a globular protein. Most enzymes are globular, meaning that they're roughly chunky as opposed to being, spiky or cylindrical. And the reason why they're globular is that there is an active site nested somewhere in that ball of protein. And that active site, shown here in yellow, is where the substrate finds its best fits. This is the active site. The active site is where you have the magic happening. It is where the bonds of the substrate are formed or where they are broken. Now, note that there's a lot of little nooks and crannies in this 3D image of an enzyme. And that's important because that allows enzymes to be very, very specific to their substrate. So here then is a big theme of enzymes, but not of catalysts, is that enzymes are a kind of catalysts that are highly specific to their substrate. So for example, a phosphatase cannot work on a carbon substrate. It only breaks down the OP bonds of phosphoesters. And even then, we have further substrate specificity. For example, phytase versus lipase break down phytic acid versus lipids. And that is all mediated by the physical structure of these enzymes. There's also another implication that is relevant to soil enzyme assays, which is that this three-dimensional structure is being dictated by hydrogen bonding largely among those amino acids. And so things in the environment or in your lab that can alter H bonding can also change that activity due to denaturing of the enzyme. So largely pH and temperature are two key environmental parameters, but also lab assay parameters that dictate total activity that's ongoing in the field or in the lab that is being measured. So for example, on the left-hand side, we have these pH optima curves, where on the x-axis we have pH, on the y-axis we have activity. We see that depending on the kind of enzyme, we have different pH optima. So pepsin is an enzyme in our stomachs, and that is evolved to deal with the acidic pH of the stomach. So it makes sense that pepsin is most active at pH 3. And likewise for other kinds of enzymes like amylase in your saliva, that's active at pH 7. So in soils, on the right-hand side, a lot of early work actually done out in Ohio by where Chris is, found that there tend to be pH optima in soils that are specific to enzymes, but they don't necessarily always track with soil pH, which is kind of weird if you think about it. And we'll get into that in just a second. The point here, though, is that a lot of enzyme assays use buffers to control pH, and we should be giving, I think, a bit more thought on, well, what pH do we want? Is it to mimic the soil pH, or is it to mimic some other condition that is relevant to how we manage? So let's get more into the soil context. We've just talked about some of the principles of enzymes, what they are. That's true of all enzymes anywhere. But soils are a little bit different when it comes to how we measure enzyme activity. It was first in 1982, excuse me, that Burns wrote a piece pointing out that enzymes in soils are likely to be outside of cells. And the reason for this is that a lot of the substrates, what those enzymes are acting upon, are also existing outside of cells and soils. So, for example, the organic carbon is sitting outside of the microbial cell. To decompose it, it has to secrete or put out an enzyme into that extracellular matrix. So, in other words, microbial cells, plant root cells, have to secrete enzymes into the soil solution, and there the enzyme encounters polymers. It decomposes those polymers by catalyzing the hydrolysis of bonds, and then the small monomers, these little bite-sized pieces, are available for microbial uptake. So the theme here is that in soils, the enzymes are outside of the cells. This, I think, is not too different, though, from our bodies, at least from our stomachs. If you think about your stomach, it really is outside of you. So doctors talk about how starting from your mouth through the anus is technically not you, it's outside of you, because that is a tube that has full continuity with outside. So given that, or in that sense rather, that enzyme that we just talked about, pepsin in the human stomach, is not unlike enzymes in the soil. It's being secreted by the organism into the environment that contains the substrate to decompose. So Burns, based upon that theory, sketched out what does it look like? What are the possible locations where enzymes could be in the soil? And this is the first sketch. It has since then been, made up real good looking, but the basic idea is the same, and that's that there's many different potential compartments or fractions or pools of enzymes. I like this concept because we think of nutrient pools or nutrient fractions. I think we would do well to think of that same concept for enzymes in soils. There's different fractions of enzymes. Now think about when we do soil enzyme assays with a bulk soil sample, which pool are we likely assaying the most? The short answer is that we don't really know, but we're not able to discriminate which fraction is being assayed, but we can try to speculate about which pool might be most dominant. So if you look at this diagram, the living cell is central because that's the origin of all these enzymes. Notes, I'm not sure if you can see my cursor, but if not, there's the arrow there of secretion. That's what we've just been discussing, that these microbial cells, plant root cells as well, secrete out these enzymes. Then these enzymes, once they're out there in the big old world, they can have many different fates. And so they can bind to the substrate directly and then do the work of catalysis and decomposition, but they can also be sorbed to solid surfaces, which in soils are going to be clays and minerals, as well as organic matter. We can also have physical entrapment of enzymes inside of micro or macro aggregates. And there's also discussion that a lot of the enzymes that may be technically outside of the cell might be doing so in a very different way than what we just discussed. So that they may not be bound to clays, but they may be simply bound to the surface of the cell that put it out there. The whole point here is that there's many different potential fractions of enzymes, but currently our techniques are taking a bulk metric. So I think it's not unlike nutrients in soils. We now measure them by different fractions, available, not available, total. Well, with enzymes, we're measuring the total activity, not the available, but not the more active enzyme fraction. Enzymes are,
On that note, Andrew, I'm sorry, can I interrupt there just for a second?
Yeah, of course.
So you're saying that, so when we pull like a random soil sample, we go 6-inch depth like we would for a standard fertility test, and we bring that in the laboratory, the rhizosphere is going to have a higher activity of enzymes, I'm assuming. So when we're doing that bulk measurement on the total soil, is that a good indicator of what's going to be happening in the root zone, do you think?
That's a great question, and you preempted me by a couple slides here, which
I'm
is
sorry.
good. No, that's good because it means that this is going in the right direction. Chris, I think that's exactly it. People sometimes do enzyme assays to mimic the rhizosphere, so they'll use a pH, a substrate concentration that tries to get at that. You can also sample just the rhizosphere, and this brings up questions on assays. I think a lot of assays are a little bit rigidly used, and I think it's like any other technique. You can define the parameter by what your research question is. And so if you're thinking about rhizosphere dynamics and how crop roots can tap into a certain pool of organic, say P or N, then maybe it makes sense that we change up the enzyme assay to mimic those conditions. And you're totally right that a lot of the evidence that I've seen to date suggests that the rhizosphere is where the majority of the enzyme activity is happening in the growing season, or these kinetic hotspots of activity. Okay.
Yeah, thank you.
Sure thing. So I want to make a note about language here. We say soil enzymes, and that can refer to different pools. The consensus seems to be that the majority of activity that we're assaying in soils is largely extracellular enzyme activity, in part because the substrates that we're using in these assays may not be diffusing into the cells. So we're really measuring, people think, enzymes that are outside of cells. The other reason why people think this is the case is because there seems to be a lot of enzyme activity even after you kill the microbial cells. And this is all work from the 1950s at UC Berkeley, where they literally nuked soils with radiation, then they used chloroform, etc., and they still found enzymatic activity after all cells were dead. And it seems like the majority of what's present, which depends on the soil type, depends on the enzyme, but the majority of the activity in a sample seems to be derived by enzymes that are outside of cells. So the term here that I want to introduce to you is abiontic. So not abiotic, not biotic, abiontic with an N. I always get people saying, pardon or sorry, is that abiotic? No, it's abiontic. Abiontic was made up, it was coined by Skajuns and Burns in '73. And they proposed it because abiontic, the Greek roots would imply that it is of formerly living origin, but it is no longer itself living. So in other words, it's a zombie, is how I'd like to think of abiontic. Neither living nor dead, though it came from something that was once alive. And in the case of soils, this is, again, think of the fact that these enzymes are being made by a living cell, but once they're released into the soil, they can still do their thing. They still can catalyze processes. And now they're decoupled from the cell that made them, which also makes enzymes kind of interesting. Now, there's lots of synonyms being used out there when you Google or try to find work on enzymes. They're called abiontic, extracellular, exocellular, exoenzyme or ectoenzyme, ecoenzyme. They all are getting at the same concept of there are enzymes in soils that are largely outside of cells. As I mentioned a couple slides back, the fact that enzymes are outside of cells makes them quite useful to understanding biogeochemical processes from nutrient cycling as well as soil health, but it also makes them a little bit difficult to measure. And if we look at this quote from a paper from 1973, this basically captures what I was saying about the Burns 1983 diagram, showing all the different possible fractions of enzymes. What this paper points out is that the fact that we have all these undefined locations or all these undefined compartments makes it difficult to distinguish the various contributions of these different fractions. That's the very last line here. So again, we're measuring totals, and the good analogy would be with nutrients. Chris, if you could only offer your clients total and total P, it'd be a little bit tricky to make decisions on what to apply. So that's where enzyme assays stand today, where we're working with a total value, and it might in some cases be muting or masking the active pool that is really driving nutrient cycling then and there. Okay, so here's the conceptual diagram that I like to use when I think about enzymes. We've got on the left-hand side the biotic origin of these enzymes, which is the cells, bacteria, fungi, plants. Keep in mind that enzymes are gene products. And so there's reason to think that if there's perturbations to the soil that impact the microbes, that could be management, it could be climate or weather, and that will conceivably translate to a change in the expression of these enzymes. They're then secreted, that's why they're abiotic. And once they're outside of the cell, they have different fates, including interacting with the abiotic component on the right-hand side. That includes things like minerals or like organic matter. So enzymes can be immobilized, meaning that they can be sorbed or physically entrapped by these clays in organic matter. They're also sensitive to abiotic drivers like temperature and pH. Now, typically the enzymes, when they're sorbed, they have a very low activity. The reason for that is that when they bind, they have a conformational shift. And as we discussed in the principles slides, when you change the shape of the enzyme, you change how well it can hold on to the substrate and thus how well it can perform its function. Now, what we're going to focus on for the rest of this morning is what is shown in the red box, because that's what we're trying to measure. And that itself is because, well, that's the promise of enzymes to soil science and to soil health, is that enzymes are the gatekeeper of nutrient decomposition or the availability of nutrients from organic matter pools. Again, they're the catalysts of this process. So they catalyze the mineralizing of organic nitrogen, sulfur, phosphorus, and carbon. Let's talk about the methodology, and let's do this in two ways. I want to first talk about the principles of how we measure enzymes, and then get into what are some soil-specific considerations. So since we don't really have a show of hands, I'll put this up as an example of an enzyme assay that many of you may have done already or have seen done, but not have realized that it was an enzyme assay. So this is the very popular tongue-in-cheek Soil Your Undies soil health test. I don't know if this began as a joke or if it was serious, but it's a fantastic idea. It's been proposed as a pretty do-it-yourself way of gauging relative decomposition rates of your soil, and thus as a proxy for soil health. The idea is that this gets at that process of, quote, general microbial activity, for which enzymes were proposed in the Federal Registry as an indicator. So I put this up here because if you think about it, the decomposition of cotton underwear is really an enzyme assay. What is cotton made out of? Well, cotton is made out of cellulose if you dive into it. And if you think about then what is doing the decomposition of this underwear, of this cotton, as I've said, microbes get all the credit for what enzymes do. It's really not the microbes, it is a specific enzyme. That enzyme is known as cellulase. So it's a cellulase enzyme assay to vary cotton briefs and then to dig it up and by difference calculate the amount that was decomposed. So this basically is an enzyme assay. Enzyme assays are always measured as a rate, so their amount of substrate per unit time that is decomposed. If you buried those underwear in the spring, a couple months go by, you could actually derive, I think, a pretty defensible metric of a rate of cellulose decomposition. And so that is why, or just to give you one example, people have looked at the activity of cellulase by itself in response to things like tillage types. And the response of this enzyme's activity, meaning in this case it's higher under a no-till or under a conservation till, likely due to more residues being kept on the surface. That's consistent with the role of this enzyme. So as a side note, it'd be a pretty cool study to compare the activity of cellulase assayed by underwear versus by the substrates that cost a lot of money.
And on that note, something kind of interesting to go with that is there's a lot of interest and a lot of people working on biological amendments to deal with residue. So sometimes too much corn stalk is going to cause a problem. You want that to break down a little faster. So with people that are using products to help break that stuff down, it'd be interesting to see what the cellulase activity is in that soil and how it's impacted by some of those biological amendments to like they say, if you're adding a lot of soluble phosphorus, you can kind of limit your phosphatase activity. I'm kind of curious if we're using some of those biological products, are we also in turn then going to be sort of limiting some of the cellulase activity in a situation like this?
That's a great point, Chris. Yeah, so I've got a few thoughts there. Yes, it seems like a lot of biostimulant products, as they're currently being called, they involve the direct application of enzymes to soils at a pretty low rate. And people, I think, are a bit skeptical of that for a couple of reasons. One is that how can just a couple liters per acre of an enzyme product really lead to an appreciable breakdown of, say, corn stalk? Well, one, again, as a catalyst, you don't consume that enzyme, right? So it's not like adding a fertilizer that gets used in terms of a end source. So there's reason to think that adding enzymes to soils as an amendment could help things with breaking down, for example, stover and, say, continuous corn, excuse me. But there's a second reason why people might be skeptical of these additions of enzymes as an amendment, and that would be that it's a really dog-eat-dog world in soils. If you think about what enzymes are built of, they're full of nitrogen, right? They're made of amino acids. And the C to N ratio is about 3 to 4. Because of this, there's a dog-eat-dog world of soil enzymes where other microbes are typically hunting down and breaking up enzymes that are being secreted by other cells. And this is done by a whole class of enzymes called proteases, which are these generalist, non-specific enzymes. And proteases, their only job is to break down other enzymes. So the skepticism of using these enzyme products in agriculture is that, well, they won't last very long out there because they'll get eaten up by a protease, and that nitrogen will be taken by some other cell. But I think this is a very, very rich area because we do this with vaccines. We can stabilize enzymes and proteins onto minerals to protect them from proteolytic degradation. So here's where formulating enzyme products might come into play. Can you formulate these enzymes to be protected from the protease breakdown? So I think there's a huge world here where agriculture and soil science can learn from, say, biofuels, where they work with the situation, or from vaccine science on how can we add enzymes to a complex system, keep it going, and have it do its function.
Would it be change? Well, if you change the shape of the enzyme, then you pretty much change its whole being. It's I don't know. That's a very interesting. I'm sorry, I don't mean to take you off track.
No, and so then this is like this is the game of ping pong, right? So that's a great point that you raised, and then it would ping pong back, and then the answer might be you're right. So there is that tax of activity. There's a penalty, and that penalty when the enzyme binds to a surface is pretty big. We found up to 95% inhibition of phosphatase just by binding to iron oxides. But what if you could engineer enzymes to be stable and active at the same time? So here's where bioengineering could play a big role in purifying enzymes that are designed to be active when bound to, say, light or kaolinite. So I think that's really a rich field for engineering enzyme products for soil nutrient cycling and soil health. Okay. So in the methods, I want to talk about first, before you do the assay, there's already some practical questions to answer for your labs or for your clients. And that would be, how do I take my sample and how do I store it, most importantly? This is a pretty contentious debate. And I think here we have had a lot of importation from microbiology of soils, namely people who are used to measuring phospholipid fatty acids or genes or microbiomass carbon In these cases, we want to preserve the field fresh status as much as possible, right? We want to capture in situ what the microbiomass composition and quantity is. And in the case of genes, if you so much as sneeze on the sample, they can change. Now, enzymes are not inside of cells, and so their fate is largely decoupled in soils from the living cells. This might give us more room, practically speaking, to not have to worry too much, or as much rather, about from the time of sampling, from moment of sampling, how much time can go by before you perform the enzyme assay. This debate really boils down to really two things. One is, what are you trying to measure? Is it a relative indicator of changes? In which case, if you air dry, can you still preserve a relative difference in, say, no-till versus till? And then the second question is one of the trade-offs of practicality of, do you want to do all the extra work and cost for running fresh samples versus maybe sacrificing a bit of what is going on in situ, but being able to scale up in the quantity of assays? So let's talk about that. I think the general consensus from papers that have been published starting in the 1970s is that, in general, when you air dry a sample, or if you store it, even if it's at 4 Celsius or frozen, there's going to be a drop in the activity of most enzymes. And I say most because a lot of these different questions on sample storage, on how you do these assays, it typically depends on the soil and on the enzyme, which is just what we want, right? For it to be very context specific. So there's no universal answer or consensus on what's the best for storage, for assay conditions. But in general, for most enzymes in most soils, if you air dry or keep on ice for more than a day or two, it seems like the activities drop. And on the right-hand side, that figure is one of probably 50 or more papers that show this. Now, I think here we have some room to think about what the research question is, and on that trade-off of speed versus preserving the in situ activity. So let's talk about whether or under what conditions air drying might make sense and when it might. And I think one here would be the climate type. And especially for the southwestern part of the US and in the tropics, soils undergo long periods of being dry. Maybe not air dry level, perhaps at the surface soil in a desert, yes, or in the tropics during the dry season. So in that case, since soils already experience in those regions air drying, you might want to argue that if you let those same soils, when they're wet, go back to air dry status before you run that assay, it may not be a big deal. On the other hand, if you're studying wetland soils, maybe air drying is good to always avoid. So I think it's important to keep in mind what the context of your soil sample is when you're doing these assays. I think it's a definite that air drying helps speed things up. And it seems like it also might increase the accuracy, because if you think about weighing out 1 gram oven dry basis of a sopping wet clay-rich soil, it's hard to homogenize it when it's wet in a way that prevents little hot spots coming up in that 1 gram oven dry mass basis. We found this for our lab that the CV increases if we have more clay content and more moisture content. So in these cases, being able to air dry and then even to sieve to the finest fraction can just homogenize the sample. It also makes the weighing out much more quick. And this all translates to more high throughput. So again, these are trade-offs. And my point is that there's no real answer. I'm hesitating here because I could talk about this for probably 5 hours, but how about I pause? And Chris, are there any questions on this question of sample storage?
I haven't seen anybody put anything into the chat window. I went ahead and just opened up everybody's microphone, so feel free to chime in if you do. But what we've been doing here, what we're planning to recommend to our consultants when things go on, is with the four assays that we'll be running for the enzymes, we're air drying here, and trying to run it within a week of sampling. So, but we do have a policy here that we hold all samples for a minimum of 90 days, so we can go back and do a recheck if necessary, but that's usually for fertility. I really have no idea how that's going to hold if we look at the same soil sample for an enzyme, 3 months later. I'm assuming it's going to look just like that graph you've got shown there.
I think so. From what I've gathered, it seems like that a lot of times when you store, be it air dried or be it in the fridge at 4 Celsius, there's a rapid drop off that's even within a couple days. We found this for Midwestern systems, that even if we run the enzyme within 2 hours, and so here the student has to sprint back to the lab on their bike literally and run the assay right away. If we look at the same soil sampled the same day after 8 hours, we found a third drop in enzyme activity. But then that's stable. I think if you can't run it the same day, it might just be worth waiting up to a week and to not kill yourself, is what we're gathering. And that's the same we found for 4 Celsius versus air drying. So Chris, what you're describing of maybe air drying to standardize and then running it at a certain time, but always keeping that time constant, I think that's a good approach.
Yeah,
one thing that I wondered, oh, sorry.
Well, and with that technical note, the technical note that you referenced there, NRCS released last September, that's kind of driving what we're doing here at Brookside on going that direction. It kind of alluded to that of all the hundreds of enzymes that were proposed that could be good indicators, those particular 4 were selected because they represent carbon, nitrogen, phosphorus, and sulfur, but then also because those particular enzymes, from what I gather, and I could be wrong here, but you can kind of couple with a standard soil sampling, and the requirements for storage and shipping and things like that are minimal. I mean, there's always going to be a trade-off and a change, but those particular enzymes maybe are potentially less affected by storage and time as others. But on that note, though, there's also, from what I gather, there's a second technical note that's being put together by some folks that's going to directly reference some of the sampling questions to go with the first note on these are the indicators, and a second technical notes being formatted to address sampling protocol.
I was not aware of that. That's good to hear. Huh. Well, on that note, Chris, also, you just said, can you use the same soil that's being run for other things for enzymes? And I think that's really key for making enzyme assays more widespread. If there's a whole different pre-treatment procedure, that's going to limit, for example, a commercial lab, how they can integrate that, or at least the affordability of it. I want to point out that I think a lot of the soil health tests, like the Solvita or the CO2 burst, etc., I'd like to propose that might be a way to build in enzyme assays where you could run the enzyme assay at the end of that incubation, be it a 6-hour, a 12-hour, even a 48-hour. I know that it differs. Dr. Franz Lubbers has looked at different lengths of how long do you incubate before you measure CO2 burst. And if you think about that is effectively a re-wetting treatment of an air-dried soil. What if we were to measure enzymes on that soil at the end of the CO2 quantification? That could be a way of, of trying to mimic perhaps a standardized field moist condition of these enzymes. Just a thought.
That's a great idea, Andrew, and we'll run some next week. We'll follow that discussion up later, but that's a great idea. We'll run some here and see what happens.
Cool. Okay, so moving on, I just want to present this table from a recent paper, well, 10 years ago now, just to give you a flavor of how much the effective storage, either it's air drying or frozen, how much the effective storage method interacts with the soil and with the enzyme type. And so in some cases, you see a tripling to a halving of activity, but that depends on the enzyme and on the storage method. So again, this is what makes enzyme procedures really quite challenging, is that it's hard to dictate a blanket best practice across even for a given soil type for all enzymes and vice versa. So let's talk more about assays. I want to walk through the basic steps and then consider the different parameters that have to be selected for each step. So the 4 steps of any enzyme assay, be it human biochemistry, aquatic systems, soil systems, is to always subject or to expose that enzyme in the sample to your substrate, which makes sense. And typically with purified enzymes, so one single enzyme, this is done by using a buffer that can maintain a defined pH. And that pH is the pH of the original environment of the enzyme. So again, this is most common in human biochemistry where people are purifying enzymes outside of the cell context. And then they try to mimic the cell pH by using a buffer. We'll talk in a second about what that means for soils, which again, the enzymes aren't in the cells in the first place. The second step is then you incubate this mixture at a defined temperature. And typically that temperature has always been the temperature of your sample, the original sample type. Third step is that you stop that reaction. So you halt the enzyme, and this is typically done by poisoning the enzyme in a non-specific manner. So using high pH is most typical because again, high pH will denature most enzymes, just as low pH will. And in soils, we use a high pH to stop that reaction. The final step then is that we quantify the amount of substrate that was catalyzed into product, either by either as a substrate that decreased or as the product that formed. And in soils, we typically quantify the amount of product that was generated by the enzyme. So here's what that would look like in a little bit more detail. On the top, I have an example of a commonly used soil enzyme substrate. This is for phosphomonoesterase, oftentimes called phosphatase. So the monoesterase is a bit more specific than phosphatase because it gets at the fact that we have different kinds of phosphatases, those that cleave the COP bond and those that cleave a COPOC bond. So whether it's a monoester or a diester bond. So on the left-hand side, we've got the substrate that we're adding, which is para-nitrophenyl phosphate, a very commonly used colorimetric or chromogenic substrate, which means it turns a different color when you make that product. On the very bottom, you have the step for the soil assay. So we combine the substrate with soil, typically in a buffer. There's enzyme in that soil, so that enzyme encounters the substrate. It will hydrolyze the bond. You generate the product, or one of the products, which would be the para-nitrophenol with an OL. That phenol at a high pH turns a bright yellow color. So that's why we terminate these soil enzyme assays, both to halt the enzyme by denaturing it with high pH, but also because the high pH changes the phenol into a yellow color. And we can quantify that as absorbance at 410 nanometers on a UV vis. So from this, we get the activity, often called the potential activity, which is technically a rate. So enzymes, they're measured as rates, which is the amount of substrate, so the micromole of PNP that is hydrolyzed, i.e. the amount of product formed per gram of soil per hour. So there's a time component. This time component, I think, is worth pointing out, is one parameter that is often assumed, but we might want to think about playing with that more. Typically, we use 1 hour incubations where we'll put the soil with the substrate for 1 hour. You see 2 to 4 hours for polymers like cellulase. And this is because these big polymers tend to take more time to break down. So you don't build up enough of that product to quantify if you only use a 1 hour assay. On the flip side, I think that some of our assays of the monomeric or these end state steps, so going from phosphomonoester to phosphate, catalyzed by phosphomonoesterase, those kinds of terminal step reactions, they happen so quickly that they might be shortened to 30-minute assays. And again, because you can just simply divide by the exact time when you're calculating the activity, there's reason to think that you could expedite the throughput of these enzyme assays by changing that time component. So here's where there might be some methods work done for any given lab to decide, for us, does it make sense to shorten or to lengthen the duration of this assay? Note that assays are endpoint assays, which means that we're not measuring the live or the real-time generation of that product, as is often done in biochemistry. Excuse me. We're starting the enzyme, we hit the stopwatch, then we halt that enzyme assay, and we quantify the amount of product, and we divide by the amount of product generated, then we divide the product that is formed by the total time. And in the case of soils, we also divide by the mass of soil. So time is one component of your assay. The other components would be pH and substrate concentration, as well as temperature. So let's talk about pH. We know that most enzymes in any sample, be it the human stomach to soils, have an optimum pH, meaning that they're most active, they're most able to catalyze, or they're most rapidly doing so at that pH. And a lot of the early work on soil enzymes found that for a general class, like say phosphomonoesterase or phosphodiesterase, for a soil type like mollisols, there seems to be a pretty well shared or preserved pH optima. What you're looking at here is one of the first pioneering papers on pH optima of soil enzymes looking at phosphatases. And what they found was that in acidic soils, you tend to have acidic pH optima shown on the left, and also the same is true of alkaline soils on the right-hand side. So this led people to believe that, well, the hypothesis that enzymes in soils are evolved for the soil pH as their enzyme pH optima is supported, and that makes a lot of sense. Microbes would evolve their enzymes to be most optimum in their environmental pH. Now this is for bulk soil, so bulk soil pH might be different from the rhizosphere pH. This has then implications for maybe you might want to change the pH of your assay if your sample type is different or if your question is different. Also think about how things like liming, when you change the pH quite violently, right? Liming is a sudden change of pH, how that might impact enzyme activities in your soils and what that means for your assays of your soil enzymes after liming. Now, most enzyme assay procedures have a codified, a canonized optimum pH. And a good example is that of the phosphatase family, so the phosphomonoesterase, the phosphodiesterases. And we have bracketed here those two enzyme classes, and on the right-hand side, what we typically recommend as a community that people use for the buffer pH. So acid phosphomonoesterase is typically acid at 6.5, the alkaline one at 11. And this goes back to the work by Enzyme along with Tabatabai at Iowa State. And you can see 6.5 for the phosphomonoesterase and 11 for the same enzyme, but in alkaline soils. Hence, we have alkaline and acid phosphomonoesterase. And then for phosphodiesterase, it is thought to be 8. Note that for other enzymes, be they for carbon, nitrogen, sulfur, there are typically more acidic pH optima that are proposed. So these enzyme activities are being used by everybody that run enzyme assays, regardless of their soil pH. And I think this raises some interesting questions because you may have some soils that don't have the same pH as the recommended pH optimum. The question there becomes, do you still use the standard pH that is recommended for the assay, or do you use your soil pH? So there's reason to think that what we think of as the common pH optima is not really the case. So on the left-hand side and on the right-hand side, you have respectively phosphomonoesterase and diesterase. And we see in the dotted red lines, the oft-proposed pH for the buffers. And in some cases, they align well with, in this case, the soil enzyme pH optima. For example, on the left-hand side here, we have the pH optima for phosphomonoesterase at around 5. So it's not that well aligned with the 6.5 that is commonly thought. but for the alkaline activity optimum, it is right on the dot for 11. For for the phosphodiesterase, we see that for one soil type, the one that's under ag systems, pH 8 is the optimum, but for soils under grassland and forest, it seems to be more acidic, around 6. So I bring this up because we've known for quite a long time, since the 70s and the 80s, that this table of recommended pH values for enzyme assays are often assumed but not always tested. It might be worth looking at if the pH optima of your soil is in fact aligned with what is being recommended, right? Now that's a different question from what your soil pH. So you could have mismatch between the recommended assay pH, what you're seeing here, mismatching that from the pH optima of your enzyme in your soil, and then a further tier of mismatch would be with the soil pH. So it's possible to have a soil pH that doesn't match the pH optima of the enzyme in that soil, that also further doesn't match the recommended assay pH. And we've seen these in acidic soils in the tropics. This has also been published. So I bring this all up because I think buffers are really a problem. My own opinion is that we should get rid of buffers altogether. And some of you who have done assays might be rejoicing because buffers are a lot of extra work. They add cost, they add time. I think there's a lot of assumptions being made about buffers. What we just talked about, you're assuming a pH optima, you're not actually verifying that. But there's also a theoretical reason that we should abandon buffers and soil enzyme assays. This goes back to the fact that we like to copy biochemistry methods in soils from human biochemistry, but it doesn't make sense for our sample types. A good example here is temperature. The temperature of these enzyme assays is 37 Celsius. You may have wondered where that comes from. Well, that's the human body temperature. That's what these enzyme assays were developed for human biochem. 37 Celsius for soil enzyme assays may not always make sense. The same goes for buffers and pH. So the reason why we use buffers is because traditionally people have been purifying a single enzyme out of a cell, and the buffer is meant to mimic the pH buffer inside of the cell. In soils, we know that the enzymes are outside of their cell and that they already are existing in a buffered system, the soil, right? Soils are buffered systems. So we're using a buffer in a buffered system already. There's no extraction of the enzymes out of the soil sample. This is all to say that there's good conceptual as well as practical reasons for not using buffers. We're not going to risk these assumptions. We save work and cost, and we also are getting at the real experience of the enzyme in the soil. There's evidence that buffers, because they are organic compounds, can compromise the activity. So if you run an enzyme assay using a different buffer type, even if it's at the same pH, you can get different activities. And these will also differ from the enzyme activity assayed in just water. So again, no real answer here. This is an ongoing area of research, but I think to try to get at what's going on in situ, going buffer free and just using water might make sense. So I know that we're going till 10:30, correct, Chris?
Sorry, I was muted there. Yeah, give or take.
Sure. So I'll try to wrap this up in another 13 minutes to give us 10 minutes for discussion, if that sounds good.
Sounds good.
Okay. So this is not a new idea. It's not my idea. It's been argued by, again, a lot of these early papers in the 1970s, pointing out that there are artifacts of buffer type and that we might just be best avoiding the assumptions as well as the theoretical redundancy of using a buffer for an already buffered system. So that, as a side note, is a big theme of soil enzyme assays. There's a lot of old papers, from the 1970s. They look old because they're scanned PDFs. I feel like we could benefit from reading those papers. A lot of the questions that we still struggle with today, from methodological to conceptual to how do we interpret these, they were all identified by the 1960s and 70s. And we've kind of taken this technique and we've run with it as a quick and dirty indicator. And that's been very useful. But as we get deeper into this tool, I think it might be good to revisit these fundamental questions. So let's go ahead and talk about substrates now. So there's a lot of options for how you run enzyme assays. Not only can you choose which enzyme to run, you also have a decision to make on, well, how do I assay it? Do I use a natural substrate, so a compound that would actually be present in the soil, or do I use an artificial substrate? So then the second question would be, do I measure the disappearance, the consumption of the substrate, or the emergence or the formation of the product? And the way that we typically do it in soils is that we use artificial substrates and we monitor the generation of the product. The reason for this is that if they're artificial substrates, we can build in a compound that is unique, not found in soils, and thus doesn't get compromised by the high background that you typically see of the natural substrate. So for example, a common natural substrate would be to use DNA or phytate itself. You add it to your soil and you incubate it. The problem is that if you try to quantify the generation of phosphate being cleaved off of the phytate, there's oftentimes a high amount of background orthophosphate, and it's difficult to get good readings of enzyme assays. So this is why we typically use artificial substrates, and we look at the production of the product, which tends to be a smaller compound and thus more quantifiable by UV-Vis or by fluorometric techniques. So the main kinds of substrates that we look at then that are artificial are chromophoric and fluorophoric. So color versus fluorescence are the two main ways of doing these assays. In general, in soils, traditionally soil science and agronomy journals publish with the chromophores, which is the para-nitrophenol-based substrates. And the ecological literature tends to use the fluorometric assays, as well as the biogeochemical lit. Not sure why, but that's how it tends to pan out. There's increasingly a lot of work on comparing it. So Shipping Deng and Richard Dick have both pioneered a lot of the work on the comparability of these two different techniques. So let's talk a bit more about what is used in soil assays. I believe that the methods being proposed for codification by the Federal Registry are the colorimetric assays, and those would use a chromophore known as para-nitrophenol. That's the compound here. This is the product that you generate by the action of the enzyme. And key to this being used is that it turns a bright yellow color at pH 7576. So when we terminate the reaction or that assay with a base, we both halt the enzyme by denaturing it. We also generate this color. The other family of substrates are those that are linked to a fluorophore. So when you cleave the bond, you release the fluorophore compound. There's two major types, the AMC and the MUF, also called MUB-based substrates. Again, these are then quantified by fluorescence, not by absorbance. Now, in both cases, we use alkaline termination to produce a color or a fluorescence that can be quantified. The difference between, or the main difference rather, between the chromophore and the fluorophore is that for the fluorophores, you have to excite the fluorophore and then you quantify the emission of that energy. So that's why in the fluorometric approach, you always have the excitation and then the emission wave number. In contrast with the chromogenic substrates, you just measure the absorbance at 410 or 4 or 5 nanometers. There are a lot of great resources out there that are hands-on. They're video-based. One for the fluorometric enzyme assays can be found on the Journal of Visual Experiments, JOVE. This is done at the Wallenstein Lab at Colorado State, Fort Collins. They're a fantastic lab when it comes to enzymes, and I recommend this video for a real hands-on example of how to get the fluorometric-based assays going. Now, this video also talks about the plate-based method, so quantifying the enzyme product using a plate reader. That same technique can be used with an absorbance plate reader for the colorimetric enzymes. Excuse me. Now, even though we've got different kinds of artificial substrates, the point is that the same enzymes can still be assayed whether we use the PNP linkage or the MUF or MUB fluorescence-based link substrates. And again, the main compound and thus the main bond is preserved, as you can see here. And it's that artificial tag or decoy or proxy that we're quantifying that differs between the PNP, and the MUF-based assays. So now let's finally talk about some of the methods, or rather some of the corrections that our lab has been working on to increase the accuracy and the comparability of these enzyme assays. I want to start with a bit of background again, historical. The first, and I'll focus also on the colorimetric enzyme assays. So using paranitrophenol, again, this is what the NRCS-based method is considering, what the soil health methods are considering. So the first use of this PNP tag was for assaying phosphatase activity in human blood samples. That was in 1939. So that's 80 years ago, I believe. My math is off, but and it was done in German because back then all chemistry was German-based. This method was then imported wholesale into enzymes. And so I mentioned at the beginning that in 1969, 50 years ago, was the first application of enzyme assays to soils. I really should have specified that was the first use of artificial substrates. So before that, people were using natural substrates, so using DNA, using phytate, and incubating it to look at enzyme activities. The major revolution in soil enzymes was the use of these artificial substrates, and that was with the same substrate that is most common today, paranitrophenol. So again, this is in 1969. Note the use of temperature of 37 Celsius. That is directly from the method of 1939 of the human body temperature. So again, we're importing wholesale the methods. And as a result, there have been some issues with the methodology when it comes to soil-specific considerations. So the first assay using PNP was in 69. About 25 years later or so, there was a paper by a British biochemist named Malcolm. And their whole point was that in soil science, even at that point in time, already there was a disregard for a lot of the basic tenets, a lot of the basic best practices of enzymes in biochem. And if you look at the opening line of the abstract, it concludes that the basic rules governing enzyme assays have, at best, been only partially obeyed in soil science. It's a pretty damning statement that a lot of the techniques that we're using, and this is back in 1982, they're not really up to snuff. And in this paper, Malcolm lays out, here's what could be done better, and here's why that matters. And the basic takeaway is that the sensitivity to treatment effects with the current methods being used in a soil enzymes, that the wholesale importation of biochemistry techniques to the context of soils ignores some soil-specific artifacts that, as a result, can compromise the sensitivity of our enzyme data in soils to treatments. So for example, you might miss a real difference between till and no-till systems if you take Malcolm's proposition to be true. So our lab is working, following up really on what these scientists said, 50, 30 years ago. We're trying to quantify, okay, well, just how bad is it if you skip over certain corrections? And to what extent can we improve the comparability and the sensitivity of these assays to treatment effects? So for this, again, we're working on the paranitrophenol-based assays. I'm walking you through this method because we are looking at a couple specific steps of the PNP-based approach that we know might be compromising the utility of the data. So the first problem is that when we go to terminate the assay, we can get incomplete recovery, we can get interference of dissolved organic matter, or DOM, that is co-extracted by the use of a base to terminate, right? We terminate to halt the enzyme, it denatures it. We also terminate with base to increase the pH to then to enable the yellow color to develop. But as we know, base can extract DOM, what we use for humic acids. And it turns out DOM can contribute to the absorbance at 410 nanometers. So you might be overestimating enzyme activity because you're falsely attributing the DOM absorbance to the product absorbance. A second issue that comes also in the alkaline termination step is that you may not be recovering all of your product, which is again that PNP compound. And this is because para-nitrophenol can bind pretty well to soil surfaces. And it can bind well enough that it is even used as a way to quantify specific surface area of soils. And that's what you're seeing on the right-hand side. So these are both two potential artifacts of the PNP-based assays that are specific to soils. In human biochemistry, in that 1939 paper, there isn't organic matter in soil to be co-extracted. It was human plasma, blood plasma samples. And also those plasma samples lacked minerals that could absorb your product PNP. So for soil assays, we have to think about, are these potential artifacts appreciable? Are they throwing off our data? So to give you an overview of that, here's a brief study that we did to assess how bad can these artifacts be. I mean, we looked at different soil types, but also we looked at different commonly employed termination steps. So different combinations of base and different combinations of flocculant. And the most common one here I'd like to point out would be the one by Tabatabai, first published as a soil enzyme chapter by Triple Assay in 1994. We also looked at a second issue of what if you don't have enough substrate, how sensitive are the enzymes to treatment effects? So let's start with the framing of this study. We looked at, or rather we used 12 soils that are just quite different. They're from different climatic and parent material gradients in the Sierras of California. We chose these because we know that there's a big gradient of phosphorus, and we're using phosphatase as a model enzyme to look at these assay artifacts. So we knew that we'd have a big gradient of enzyme activity. And if we look at these two artifacts, we find that, not surprisingly, it depends on the soil type, how much the artifact can compromise the measured activity. On the left-hand side, we're looking at the question of the DOM that gets co-extracted by the alkaline termination. And what we find is that there are some soil types that can have up to a 3-fold overestimation of activity because the DOM that's co-extracted is being read as the product of PNP. In general, though, most different termination methods for most soils on the x-axis have about a 30% overestimation risk if you're not accounting for that DOM absorbance. On the right-hand side, we're looking at the artifact of what if you are not fully capturing all of that PNP that is being released by the enzyme. And here we find that the degree of underestimation is less variable than the degree of overestimation by DOM. And it's a little bit greater, it's about 38%. Now note the trade-off here, that if you decrease the intensity of the base, so if you go from hydroxide to tris, you tend to get less co-extraction of DOM, which makes sense, the base is weaker. But you also get less co-extraction or less extraction, period, less recovery of your product, of the PNP. So there's a trade-off between overestimating activity due to DOM and underestimating the activity due to incomplete recovery of PNP. And what we do in our lab is we use the 0.1 molar tris with 2 molar calcium chloride, or we use the 0.2 molar hydroxide with 2 molar calcium chloride as what, for our soil types, the optimum trade-off of tamping down on DOM co-extraction, but also maximizing the recovery of PNP. Okay, so let's talk about the issue of substrate saturation. I've hinted at this a couple times, and I think this might be the biggest issue. It's what Malcolm in the 1982 paper points out as the most egregious oversight of soil enzyme assays. And that's that we don't necessarily make sure that we are saturating the enzyme in the soil sample with enough substrate. To understand why this is an issue, we have to think about how enzymes operate. I mentioned that they're a catalyst and that they really are measured as an activity. So that's, in other words, it's a rate. How fast can it go? There's a point where if you add enough substrate on the x-axis, you see that the activity, the rate of catalysis on the y-axis saturates out. It can't go any faster. And we can quantify this or call this as the maximum velocity or the maximum rate of catalysis known as Vmax. Now, the amount of substrate that it takes to get to half of Vmax is your Km. So to think about Vmax and Km, you look at your Vmax on the y-axis, then you divide that by 2 on the y-axis, then you trace out on the x-axis what substrate concentration shown here gets you to that half of Vmax, and that's your Km. The reason why we talk about Km and Vmax is that in traditional biochemistry, we ensure that we approximate Vmax by using a substrate concentration that is 5 times the Km. So 5 times Km puts you at roughly 88% plus Vmax is what people have found. In soil science, this would mean that for a soil sample, we have to first make this substrate saturation curve by using increasing amounts of substrate, calculate Vmax, calculate Km, determine 5 times Km, and then go back and use that substrate for the assays. No one really does this, and that's what Malcolm was pointing out is a potential flaw in how these assays are used in soils. Here's why that might be a problem. So if we go back to our study on those Sierra soils, we've got we did an assessment of what is the risk of having enzyme assays done at sub-saturation conditions, so not enough substrate. Well, here's what that looks like. On the x-axis, we have substrate concentration, and on the y-axis, we have the activity of phosphomonoesterase. And this grayscale box is the range of substrate concentration that is used in current recommendations. So this is from the Tabatabai papers book chapter. It is a equivalent of 10 millimolar per gram soil per hour. So you often hear about 50 millimolar being added to the enzyme assay, but that's of the 1 milliliter added to 4 milliliters of buffer. So the final concentration of substrate is really 10. That's what that 10 region is that we were considering as the reference. Now we took 3 soils and we pushed them to their Vmax, and that's what you can see in these saturation curves. What we find is that we see a true difference, or rather a significant difference, come out for 3 of the soils only when we push them to their Vmax. And if we use the substrate concentration that is recommended, we would miss the significant difference between these 2 soils in the triangle and in the shaded circle. What this means then is that if we're running enzyme assays under sub-saturation conditions of substrate, we risk false negatives. This kind of makes sense because you think of Vmax as an inherent property of the enzyme. How fast can it possibly go? You need to push the enzyme to its maximum limit to really pull apart differences. So not only is there a risk of false negatives by your treatments if you use non-saturation conditions of substrate, think about the comparability of data across studies. If we have enzymes that are being assayed at a sub-saturation condition, that activity is not inherent to the enzyme. It's simply a function of how much substrate was used, which means that we can't really compare values across what's been published. This raises a big issue for meta-analyses, also for monitoring programs of soil health across space and time. To give you a sense of how much substrate concentrations vary, here's a graph that our lab pulled together. The circles just show example papers. The ones in shaded gray are more of these seminal papers, like for example, the 69 paper that was the first use of PNP assays. The Tabatabai 94 paper is the first soil enzyme chapter published by SSSA. So there's a lot of variability in these activities, in the substrate concentration. Again, keep in mind that this threshold of 10 is what is typically used for the current recommended practices. So I know we're running a bit tight on time, so I'll just end by saying that one recommendation that comes out of this is that we need to do what our lab calls a priori pains. So a priori, meaning before we do assays, and pains because it's a little bit painful to do all these corrections for DOM, for PNP absorption, and also most importantly for the question of how much substrate do I have to add to achieve saturation of the enzyme in this specific soil for this specific enzyme. That's a lot of work. Now, what we do in our lab is that for a given soil type, at the outset, we do a substrate saturation curve. We quantify DOM interference and PNP absorption. And then, depending on the research question, we do it by sample, or we assume the average values or the average conditions derived at the outset of that study. I think a lot of the reasons why we don't do these extra a priori corrections and work is that it's frankly a lot more money. So a substrate saturation curve has 6 to 8 substrate concentrations. So that's 8 times more assays that you have to run. If you then do a correction for DOM and a correction for PNP, now you have 10 times more assays to run. That's 10 times more labor, 10 times more substrate. But on the other hand, as we found out from papers like Malcolm, and as our lab is finding, if we don't perform these corrections, we really can compromise how useful the data is. So false negatives, and also over and underestimating by up to a factor of 3-fold, which is fairly problematic. So, well, 10:30, sorry for running right to time. I've got plenty of time to stick around for questions. That's what I really wanted to get through. I'm happy to hear any thoughts you have and discuss enzymes.
Well, thank you, Andrew. I'm sorry, I think somebody else was getting ready to speak. I apologize. I'll quiet for a second. Maybe that other voice I heard was just myself feedbacking from somebody's speaker or something. But on that last note there, you were talking about how much of that saturation pitfall, I guess, could be maybe on these 3 particular samples. Was that a pretty comparable texture, or was there a heavier clay or a different different clay minerals in those soils, or how different were they?
Yeah, so actually, could you repeat the first part of that? So you're asking why might there be such big differences in the saturation of these soil enzymes by sample type?
Yeah, I guess I'm kind of thinking like,
Sure.
can some of that difference in saturation be correlated to other maybe simpler or less expensive testing? Like, can a texture
Sure.
analysis kind of explain some of this, or?
Yeah, that's a good question. And a lot of these enzymes track with soil carbon concentration in general. If you do correlations, enzyme activities on the whole have typically significant, at least at P.05 levels, correlations with soil carbon, or say with labile carbon like POX. You
Okay.
interpret that as labile carbon. And so that might be one way of framing it, that the higher the organic matter content in general, you might want to use more substrate. But we keep in mind that the amount of substrate is not necessarily how active the enzyme is. So this is a difference between Vmax and Km. Vmax is the activity that you measure, but the amount of substrate that you need to get to Vmax is dictated by Km. And they're related, but you can decouple these. So how active the enzyme is not the best way to predict how much substrate it needs to get to that activity. So Km is a measure of affinity for the substrate, not how much activity there will be in the end. What this means is that you may have an enzyme with the same Vmax, but the Km could be different. And as a result, you need a different amount of substrate to achieve the same Vmax, the same activity at saturation. So that's the flip side, Chris, is that there are proxies, but there's oftentimes no correlation between the amount of substrate that you need, which is the Km value, and the saturation level. And in fact, in this paper, we didn't find any correlation between soil carbon and the Km, and thus the substrate requirement.
And the soil carbon, as you're saying here, is that total carbon or epoxy or something like that?
In that case, it was total carbon. Yep.
Total carbon.
In
Okay.
fact, I have let me pull up another talk where I've got a slide on the same soil type. This is from the U of I has some campus farms, and so we know the soil types. Oh, wrong slide, excuse me. I want to show you an example of you can have the same soil properties, Chris, pretty much the same carbon, the same clay content, and still have a big difference. Can you see this now?
Yes, thank you.
So this is from the same soil type from 3 land uses of forest, grassland, and then corn, soy. It's on our state soil type, the Drummer series. These
So you're
samples.
mollisol here, so organic matter, what, 4% or so?
Yep. I think, yeah, 4%. From forest, it's more like 6-ish. Grassland's about 5, and it's about 3.5 to 4 for the corn, soy.
Okay.
These are these rich black soils that we have out in central Illinois. But the key thing is that all these land uses were within 50 meters of each other. So the soil type is identical, climate's identical, clay is identical. Small differences in organic matter, they're all pretty high. But look at the differences in the KM and in the Vmax. So the Vmax is how active it is, and that's what you see as this saturation out. Now the KM would tell you how much substrate do you need to get to that. So this is interesting because consider, for example, the grassland and the corn soy. There's twice the activity, the Vmax, in grassland versus corn and soy, which makes sense. But the KMs are not twofold different. So you would need a similar amount of substrate for grassland to corn soy to reach a twofold difference in the inherent activity. Does that make sense?
Yeah, thank you.
So this is why, because Vmax and KM are decoupled, you can't necessarily say, well, more organic matter correlates with more activity, but that's where it stops, because then the correlation with KM may not be the case.
Okay, thanks. And Andrew, I'm not sure if you can see the chat window here, but I've got a couple questions that have been posted. I don't know if you've got that in front, or would you like me to read them to you?
Could you please? Yeah.
Okay, so we've got first question here is, what do you think is the most cost-efficient method of determining the saturation point?
Well, you can't avoid the fact that you have to simply use more substrate. And if you think about the cost of your substrate for these saturation curves, it's these, if you can see this slide, it's the high-end concentrations that eat up the money. Because when you're running 1 or 2 millimolar and then you're going up to 50, it's doing your triplicates of 50 millimolar that are the majority of your substrate. So that's unavoidable. In terms of cost savings, I think labor is where people can cut down on costs. And here's where the well plate method for quantifying the color or the fluorescence can really help. If you can well plate in with a multi-pipetter the different substrate concentrations as well, for the fluorescence-based assays at least, that can also cut down on the labor costs. There's no real good answer here. At the end of the day, you still have to use a lot more substrate than you typically would. We have 2 ways of doing this. One is that if we don't want to invest in the substrate saturation curve, we use a safe amount of substrate, which is typically going to be on the high end. And if we run that soil type for a lot of assays, to make sure that we're not oversaturating and thus wasting substrate with the safe amount, then we get the curve empirically for that sample type. So I think there's a lot of play here in the opening stages of any soil site or project or lab. I think getting a handle on what do you see as a typical range of KM and Vmax for your sample treatment types, that might be a lot of front work. And then from there, we can provide recommendations, at least internally. And I see this may be a way forward for soil health internationally and nationally, is getting a handle on what Chris said. Are there ranges of carbon and texture or combinations of those where you can kind of bracket for a given enzyme type? If you don't want to do the substrate saturation curve, what should your safe amount of substrate be?
Thank you. And then we've got one more here. You mentioned that in your laboratory, you have a couple different approaches. And so what is the determination criteria between using the tris or the sodium hydroxide? Is it based on humic acid or organic matter interference?
It's based on a couple things, on multiple things actually. So one is that we get a lot more aggressive extraction of DOM, and so it depends on your soil type, but we found for andesols, we get tremendous DOM co-extraction, and that's why we see that 300% interference. You almost get what looks like a humic acid extraction black. So if you've got andesols or soils that have a high amount of soluble organic matter, then the hydroxide step is problematic. That's why we use tris. There's two more reasons why we use tris. One is that the hydroxide can artificially hydrolyze your substrate. So we found that for some substrate types, including the phosphodiesterase substrates, if we use 0.5 molar hydroxide, it will cleave the bond. And so you're making the product of PNP artificially without the enzyme. So we call this abiotic hydrolysis of substrate. This has been documented, but since not too many people run the diesterase enzyme with PNP, I don't think it's been identified as an issue. And we found it also the case to be true for some of the carbon enzymes like the chitinase, that if you terminate with 0.5 molar, for some soil types, and we're not sure why this is, for some soil types, using a 0.5 molar hydroxide termination will artificially break the substrate, and so you get a bright yellow color that you falsely attribute to your substrate. We also found that to depend on the manufacturer, so who we buy it from. There traditionally has been two main producers, Sigma and BWR. Now there's Acros Organics, there's Beantown Chemical. We find a lot of variability from manufacturer to manufacturer on solubility and on this artifact. The third reason why we use tris is because the whole point of using a base is to increase the pH, to denature the enzyme, and thus presumably halt the reaction, and to produce a high enough pH to make that yellow color. But tris, it's not so good at increasing the pH if you have if you have well-buffered systems, right? Tris is a weak base. So we've been moving towards a buffer-free approach for our lab. And in that case, using hydroxide to increase pH is overkill. If you don't use a buffer, you can achieve an alkaline termination using tris only. That's why we also use tris termination. Does that answer the question? I hope.
We got a positive response there. Yes, thank you.
Okay, sure thing.
All right, well, if no one else has any comments or questions, I think I'd just like to go ahead and wrap it up and thank Andrew again for taking the time to speak with us, and I appreciate you doing that, and everybody else too. It's a nice Friday morning. It's beautiful out, so thanks for joining us.
Great. Well, thank you everybody for your time.